Yes, thank you very much for inviting me and for the very kind introduction. And I'm sorry that I couldn't be there in person. So I'm going to talk about three projects today that I've been involved in some way in Google. Both a project I was involved in medical imaging that I think is a good example of how we should approach machine learning and AI in when we're designing for clinical practice. A project predicting gene expression from my work in the genomics team in DeepMind. And then although I wasn't directly involved in AlphaFold, it was part of my daily life when I was in DeepMind because so many people around me were working on it. And I've been asked to talk a little bit about that as well. So first, in medical imaging, and the piece of work I'd like to talk about is triage recommendations for patients with eye disease using optical coherence tomography scans. This was a collaboration with Moorfields Eye Hospital in the UK, and it was the first piece of work I worked on when I joined DeepMind. So OCT is a kind of a 3D scan of the retina, of the back of the eye. And it sort of essentially creates a 3D resolution, so a high resolution 3D scan of the retina. It works a little bit like ultrasound, but with light. And it's important because a large number of pathologies in ophthalmology are diagnosed using these scans. And so here is just an example of what you're seeing. So in the fundus image, that's the big red circle, that's a picture of the back of your eye of the retina. And this area centered around the macula where there's the highest concentration of photoreceptors is the OCT scan. And essentially the challenge here is that although there's a large number of pathologies that are diagnosed from this, and patients will receive these scans as an outpatient, there isn't a sufficient number of experts in the UK to report these. And so without an appropriate triage process, many patients can be waiting long periods of time when they have conditions that would actually be sight-threatening and could ultimately lead them to lose their sight within days or weeks. And so what we want to do is to be able to triage all of the scans for which patients are having these into these four categories, from urgent that need to be seen in a matter of days to those that don't really need to be seen, which we term observation only for the paper. And so the framework we proposed for this was a two-stage architecture. So in the first step, we have an ensemble of segmentation networks that take the OCT scan and produce a segmentation and essentially multiple tissue map hypotheses. So in the second step, we have an ensemble of classification networks that predict the diagnosis probabilities and provide a referral suggestion. The segmentation model is trained with about 900 segmented OCT scans. The classification network was trained with about 15,000 tissue maps with confirmed diagnosis and referral suggestions. And we compare this in the paper to a standard black box network. And we think the reason to do this architecture, or the reason we proposed this for that paper, was two particular advantages. So the first was that we felt the segmentation map provided an important clinical insight and into how the network is actually interpreting the image at every position. And so this was essentially seen as clinically interpretable to the physicians that were working with it. And I think, regardless of what models we're using, this is a really important thing to consider when we're designing for clinical practice, because the difference when you look at, when we've done multiple reader studies with different UIs and different presentations of the same information from a model, that those differences can make a huge difference to the ultimate clinical use and how humans are interacting with the model. And so we found that this was useful when we were working with ophthalmologists as part of the paper. And so we found in most cases this was sufficient for a clinician to understand the network decisions. It's also scanning device independent. So one of the challenges is if a new model of imaging is rolled out, or a new an update to the existing OCT scans, which happens every 5 to 10 years, so pretty regularly. Once that rolls out, there's insufficient data available on that new scanner to actually retrain a classification network. And we found that being able to, by taking this two-stage approach, we only needed to train the first segmentation step without actually needing to retrain the second step, and thus we think it acquired fundamental knowledge about the human eye anatomy and its pathologies. And finally, we showed the framework reached an expert performance. For the segmentation, we used an ensemble of 3D units. And the sort of result that we got is shown here for a single case. It's a relatively nice segmentation. You can also see that it's very easily interpretable to even the layperson, that where it should be entirely green, which is the retinal cells, what we actually see is this large area of edema, which is in blue, and the new vessel growth, neovascularization, which is associated with sight-threatening diseases below. And we also found that one of the interesting points of having this ensemble of models is that we were able to cope with ambiguous images more easily. And what we found is because it's unclear from this image exactly where the boundaries are and exactly what each part is, the different models within the ensemble were all producing slightly different segmentations, which actually reflected the different segmentations on the right here from the different experts. And again, this is sort of a bit more, adds an additional layer of interpretability to this model. The classification network was 3D DenseNet, and we sort of end up then with these 5 segmentation models, 5 classification model instances per segmentation, ultimately 25 different predictions for each diagnosis. And again, we can see the fluctuations in the confidence actually sort of reflecting a confidence score in how likely the model is to be correct. When we're evaluating, it's always important to get as clear a ground truth as possible. And so what we did is rather than just relying on the individual diagnoses from the CT scan, we wanted to get a ground truth that included several years of follow-up, so that we could be sure that whatever we were seeing in the scan was confirmed and had not sort of subsequently changed if a diagnosis had been either mistaken or had subsequently changed as a result of additional testing. So when we're evaluating it, this is the ROC curve for the model on the urgent referral task. We can see it's very nice AUC of kind of over 99%. And sort of to add, but this is relatively meaningless without knowing the human performance. So to do this, we gave the humans, so 8 different human experts, including several world experts, the same OCT scans and asked for their referral decision. And so this is what we got. So only the two world experts are experts 1 and 2. They were on par with the network. All of the others were worse or significantly worse. But this was also a slightly unfair comparison because experts are not normally used to diagnose OCT scans. And in actual clinical practice, rather than just have the OCT, what they're likely to have is additional imaging, and then additional information, things like the age or the sex of the patient, their visual acuity, so how well they can see, and then a little bit as well about why they might have come. And that information can help the experts improve significantly. And so this is done before that. When we gave them the additional information, we found that they substantially improved. So here, 5 of the 8 experts performed at the same level as the network, but none were superior. And so again, we can show this. This is the results for the whole, every referral class rather than the previous graphs, which were just for urgent. And we can see, so in the blue here, the light blue is the significance boundaries, so which is a significant difference. And we can see again that the 2 experts were similar, were non-significantly different to the model, whereas the other 6 were significantly worse. That was when they only get the OCT scan. And again, we can see here with the additional information, 5 of the experts were on par with the network. The other 3 were inferior. We can also look at the confusion matrix and compare that to experts. Again, the important thing, something is extremely important when we're designing for clinical practice, is to try and reduce harm as much as possible, and to develop models that are not going to lead to harm. And what we can see here is that this circled red is basically cases where a condition that should be referred urgently, that's potentially sight-threatening, is being classed by the model as observation, i.e. it doesn't need to be seen. And there were no cases of this in our model, and similar in the 2 experts, no cases of that either. We can take that concept further, so we can assign a penalty matrix based on the potential wrong decisions and the impact of that, and then we can score the model and the experts on this. And again, we can see our models by using this score, which is kind of related to how harmful a particular decision would be. Our models have the lowest score, and many of the experts are making potentially more harmful decisions as well. And finally, I wanted to look at the generalization to a new scanning device. So again, we said that there is this problem with a new scanning device. So this is a device that the model wasn't trained on. If it becomes available, the scans which are very different to the scans the model was trained on break the segmentation network. Obviously, this is looks terrible, and the performance is also terrible. So only 46% error rate. So a bit of a disaster. If we retrain the segmentation network with only 150 additional images, which is quite reasonable to collect for a new scanner if it's being released to the market, we found that the original classification network with no additional new training maintained its performance on a ground truth test set from this new device. And so we found that that was potentially useful without needing to kind of generate a large amount, thousands of new cases for each additional new scanner that emerges. So I'd now like to switch tact to something very different. This was, after I worked on medical imaging, I moved to work on genomics, and I think this again is a really interesting area, and something where I think medicine will increasingly revolve around genomics. So here we wanted to predict gene expression. So just as a quick overview, the way that expression works is essentially you have a section of DNA with which part of this codes for a gene. The process of transcription leads that to precursor RNA. We have introns, which are non-coding, and exons that sort of physically code for the protein itself. And then that's spliced to mature RNA, and then ultimately that sequence in the mature RNA is translated to an actual protein. Alongside that, there's a number of other areas in the non-coding DNA which are important in regulating that process of transcription. So we have promoters that are important in physically. These would be these blobs here are transcription factors, and promoters essentially attracting the transcription factors that help start the process of transcription and translation, and then enhancers are areas that are further away from the transcription start site, but that are still involved in regulation, and they exert their effect in a number of different ways. And so this is kind of particularly important because, as we've just said, around enhancers and promoters, many of the common traits and variants that we associate with conditions and diseases are non-coding. So that means they're not in the area that's actually coding for the gene, but rather in areas that are not coding, and they're exerting their effect in a more subtle way. And we kind of lack information on the mutations that actively perturb known enhancers, as well as we lack a complete understanding of the presence and the location of sort of cell-specific enhancers and the target genes. And so GWAS studies, which are sort of the large-scale studies we tend to do to try and identify variants that are associated with particular conditions, they tend to be limited thus to sort of frequent or more common variants that we can actually collect data for. And so models that could predict gene expression from an input of a raw DNA sequence can help us better understand the regulation of transcription and how it's affected by the many different non-coding genetic variants that are associated with human diseases and human traits more generally as well. So what we did is we essentially built a model to do just this. So here's the experimental setup. The model is trained, so it's a transformer-based model known as Informer. It basically inputs around 200,000 base lengths of DNA sequence from whole genome sequences. That's about, to put that into context of the enhancers that we spoke about, the model would see about 84% of the relevant enhancers. So there's about 16% of enhancers that are further away, either distal or proximal, further distal or proximal to the receptor field of this model. But it should see most of them. And the model here is trained to predict, trained on human and mouse data to predict gene expression, and then additional things like histone modifications, DNA accessibility, other things that are relevant in gene expression. but also to do this across many, many different cell types. So when we talk about gene expression, the expression of your, even though your DNA sequence may be the same in all of your cells, the actual levels of expression are very different. And so when we predict gene expression, we can't just predict it in one type of cell, we need to predict it in many, many different types of cell. And so that's just what this model does. And so as a result, we compared performance here using CAGE, which is a particular measurement of gene expression that's commonly used in this field. The model substantially outperformed the previous best model for predicting gene expression. And to contextualize this difference, this is a third of the gap between the current SOTA and two experimental measurements from the same cell type. So the goal here would be to basically be the same level of error as an experiment. To get a bit of a more intuitive idea of what the model is predicting, we can kind of arrange the model's predictions as matrices of gene expression. So in the upper image, we have examples of the ground truth that was observed, values from the relative expression of genes in our test set. And then below we have the corresponding predictions of our model, given only the DNA sequences of as an input. And we should note, of course, that the image here is a single example, but even despite that, the model predictions do closely follow observed values. And in fact, sort of the most prominent changes in gene expression in this example were kind of a change in CD4 cells due to particular cancer treatment. And that was actually captured by the informer model. So it can kind of capture some of these differences that are also kind of therapeutically related as well, which is really interesting. We can also look at what parts of the input data the model's paying attention to, and so here, kind of particularly for genes with CRISPR validated enhancers, we can kind of show that the model attention correlates with this sort of epigenetic modification. So this area we kind of center around this kind of gray area in the middle of the image, and we can see the end-former peaks in the blue here, and we can see them correlate quite nicely to the actual known enhancers in the kind of grey and black here, which again can kind of give us sort of scores of how likely a particular area is to be an enhancer, and potentially that can be useful in future work to identify new enhancers. And indeed, we can apply that concept systematically. So here is the two graphs of two different sort of libraries of experimental CRISPR validated enhancers. And in both cases, so the green here is a sort of experimental derived measure. The blue is our model, the orange is the previous state of art, and the grey is what you would get for a random. And in all cases, most cases, our model is either as good or better at predicting these, and the performance is still good quite distally as we go quite far away from the index variant where we're looking at. So again, this can kind of helps us understand or helps kind of give us confidence that the model is really learning something that is biologically relevant. So we can also kind of use the model to identify potential causal variants using the predictions. So in this example, we show essentially N-formers kind of an EQTL study, and the model is sort of a probability prediction that sort of trying to identify. So basically what we need to look at is the blue area here. That's where the model is essentially saying there's a relevant prediction on the reference genome. So that would be a genome of a normal individual essentially. And then the red down here, we can see that's the prediction on a pathological example, and we can see that the model is identifying reduced expression at this particular motif that correlates to the SP1 protein. And so the suggestion is that sort of this transcription factor binding site SP1, so altered binding there in the particular cell type where we're looking at. So in this, it was hematopoietic cells, so cells involved in making blood cells. That may be a particular cause of the pathology seen in this particular variant we're looking at. So this is this is the NLRC5 gene, which is a gene associated with kind of amyloid lateral sclerosis. And so this model is open source, so anyone can use it or play around with it, so please do. So the last thing I'd like to talk about is AlphaFold 2. This is an extremely important problem. So scientists have been kind of interested for a very long time in determining the structure of proteins, because a protein's form is thought to dictate its function. And once a protein's shape is understood, its role within the cell can be guessed at, and scientists can develop potentially drugs that work with the protein's unique shape. It's a very difficult problem. So we start off essentially with a sequence of amino acids in the primary structure, and these kind of fold in complex, difficult to predict ways. Sometimes it may have multiple domains and multiple different chains, and ultimately we want to be able to predict this complex 3D structure that may be thousands of amino acids long in a sufficiently accurate way for it to be biologically useful. That is a difficult problem, and one of the reasons why, to illustrate how difficult this problem might actually be. So here is 4 different myoglobin molecules. On the orange one is from humans, the others from different animals. They all look very, very similar visually. The shapes are very similar. The actual similarity between the sequences between the orange and the blue is 80%. Between the orange and the yellow is only 25%. So these are almost completely different sequences, and yet they still have a very similar structure. And the converse is also true. Small changes can result in large differences to their structure as well. The concept of predicting protein folding is, or we assume that all of the information is available in the amino acid sequence and the environment it's exposed to. So in theory, we have most of the information just in the sequence to be able to make a good job of a prediction. And people have been at this for a long time. So there's many different experimental approaches that have been tried. They all have their strengths and weaknesses. So here, the process of crystallography, which is very expensive, is quite time consuming, and there's multiple steps in which things can go wrong. And in fact, proteins may not even be stable enough in a solvent to be able to crystallize them. There are other approaches like cryo-EM, NMR spectroscopy that also have strengths and weaknesses. But ultimately, kind of, there's none are perfect, and they're all kind of expensive and time consuming and quite difficult to do at scale. Just a little bit about the actual theory around protein folding. It occurs inside the cell in the endoplasmic reticulum. It can take anywhere from sort of tens of milliseconds to a few minutes or hours, even depending on the size, the type, and the nature of the protein and such. In theory, the protein folding problem could be solved purely mathematically by simulating the different forces on the different atoms in each amino acid. We can do this with molecular dynamic simulations. If we calculate these forces and the positions of the atoms at every femtosecond, which is, the reason we do that is because that's the time that it takes for a hydrogen atom to vibrate, and that will be the fastest vibration. And so the longest known MD simulation, so molecular dynamic simulation so far, is about 2.5 milliseconds. And when we're saying, at least proteins would take tens of milliseconds to fold, if not hours. And these long MD simulations are extremely computationally intensive. We can see that at least for the foreseeable future, this problem isn't going to be solved by MD alone. And so that kind of leads us to CASP, where for decades teams have been coming together from around the world to try and solve a protein folding problem. And what we can see is over the sort of at least the last kind of 20 years, very little progress was made. In 2018, AlphaFold was entered and blew away all of the competition. Subsequently, AlphaFold 2 was entered again with substantial improvements and different architecture, and again, did even better. It really, and although AlphaFold wasn't entered last year in the CASP competition, all of the top performing models were all based on AlphaFold and AlphaFold's the architecture. So this seems a real breakthrough in the field. So I'd like to talk just a little bit about the model itself. So we take an amino acid sequence itself as an input to the model, and then AlphaFold 2 generates the backbone atoms and the various respective side chains kind of as per the sequence, and then assigns initial coordinates to each of the atoms. And then there's a few sort of pre-processing steps to go before we go to the deep learning part. So first we construct an MSA, so basically a multiple sequence alignment, and that's done across organisms to find structures that correspond to the sequence. And so the aim of this is to detect parts of the amino acid sequence that are more likely to be mutable, and to build correlations between these different mutations. And so the underlying idea is that if two amino acids are in close contact, mutations in one of them will be closely followed by mutations in the other in order to preserve the structure. And so we can see that idea here. So this grey chain of amino acids, when we look at the different sequence, the very similar sequences that we've collected, we can see that these two starred areas will always change together. So if one mutates, the other also mutates. And that would be required if we know that the structure is similar from all of these. That would be required to have the 3D, to maintain the same 3D structure. And so thus we can see, well, these are likely to be mutated together. And so we can kind of input that information then as a in this sort of matrix that's produced for the amino acid for the input sequence that goes into the model. Second, we also do look at templates. So we identify proteins, again, that have a similar structure, and from a structural template database, and we can construct an initial representation of structures for pairwise comparison. And again, this kind of helps the model represent which amino acids are likely to be in contact with each other. And this process of looking at templates is very common in the world of protein folding, such that the difficulty of a protein to fold has typically been defined by the number of templates available for it. And here, yes, this is similar to what we showed before. And although we did say that the similar structures can have very different sequences in general, most sequences that have similar structures do tend to have similar sequences. So this was an extreme example to prove a point, but in general, there is relevance to the template approach. And so then AlphaFold 2 takes these both the MSA and the pairwise templates, and passes them through a model that we call the evoformer, which has 48 blocks of neural networks to exchange information between amino acid sequences, and does a sort of pairwise structural characterization of the geometry. And that kind of simultaneously helps refine the model in an iterative manner. The architecture uses two transformers, with one kind of clear communication channel between the two. Each head is sort of specialized for the particular type of data it's looking at, so either the MSA or the pairwise interactions. And the model sort of also incorporates the information of kind of it's a little bit of additional information as well. And it sort of allows for the regular exchange of information and iterative refinement. So again, the central idea, as we said, is that information can flow back and forth throughout the network. Typically, machine learning models for protein folding would have only taken the MSA input and then just output some information. Whereas here, sort of at every cycle, the model is kind of leveraging the current structural hypothesis to improve the assessment of the MSA, which in turn then leads to a new structural hypothesis and so on. And both representations, sort of the sequence and the structure, exchange information until the network reaches like a solid inference. We can understand that a little bit more clearly here. So in the left diagram, we have the MSA transformer, which identifies a correlation between two columns in the MSA, each corresponding to a residue. This information passes to the pair representation, where subsequently the pair representation identifies another possible interaction. And in the right diagram, that information is passed back to the MSA again, and the MSA transformer receives an input from the pair representation and observes that another pair of columns exhibits a significant correlation, and this kind of keeps going and going to update until ultimately we've got a much more accurate representation. And so that information, after that process, is then passed to the structure module. So because that information is useful, but we still need to actually get a structure out. So the structure module has 8 blocks of neural networks to construct the final 3D structure of the input sequence. Basically, this module considers a protein as like a residue gas. So every amino acid is modeled as a triangle and representing the sort of 3 atoms of the backbone. And the triangles float around in space, and they're moved by the network to form the structure. The transformations are parameterized as affine matrices, which is a way to sort of represent translations and rotations in a single 4x4 matrix. At the beginning of the structure model, all of the residues are placed at the origin of the coordinates, and at every step of, again, an iterative process, AlphaFold2 produces a set of affine matrices that displace and rotate the residues in space. One of the kind of interesting points about the structure module is it uses a type of attention called invariant point attention, or IPA. And this is helpful because it kind of allows for invariance to translations and rotations. This is particularly important in this case because if the model knows that any potential rotation or translation of the data will lead to the same answer, then it needs a lot less data to pull it away from the wrong models, and thus is able to learn a lot more. And so then finally, this is kind of rerun multiple times, so 3 times, and the output is ultimately a refined protein structure with kind of Cartesian coordinates for each atom in the protein, and ultimately a PDB file. And so this model has been widely successful. It's been used for a range of different applications. There's a sort of short overview of a paper which I won't have time to talk about, but the combined AlphaFolds predictions with MD simulations to assist with protein design can also be used to sort of predict the impact of mutations on protein stability, again, which is extremely important in multiple areas, including fields of protein engineering and protein design. The model itself, I just add at the very end, is available through Google Cloud Vertex AI as well. So, and we're very kind of excited about the future potential of the work, either through all of the different ways by which people are using it. So thank you very much. I hope that was a kind of quick, maybe rapid tour through the various 3 of the projects that I've been aware of or involved in. I'm happy to take a few questions if we have time.